Welcome back to the deep dive. Today we're going to do something that feels a little bit illegal.
Just a little bit.
Not in the judicial sense. We aren't going to advise you on how to rob a bank or anything, but in the biological sense.
Yeah.
We are going to break the fundamental laws of physics, or at least the laws of life, as we were all taught them in high school biology.
It really does feel like a violation of the rules, doesn't it? I mean, usually when we sit down to dive into a topic, whether it's a virus like rabies or a bacteria like staph or even a parasite, there's a blueprint.
Right.
There's a logic to the enemy. There is DNA, or at the very least, RNA. There's a genetic code that drives the infection.
The central dogma. We all learn this. DNA makes RNA, RNA makes protein. That is the arrow of time for biology. Information flows one way. You need a code to build a soldier. Today we are talking about something that has no DNA, no RNA, no genetic material whatsoever. And yet it spreads, it evolves, it kills.
It is the ultimate biological anomaly. We are talking, of course, about prions. And to guide us through this strange landscape today, we are relying heavily on two absolute heavy hitters in the medical literature. We're using Murray's Medical Microbiology.
Specifically Chapter 56, right?
Exactly. Chapter 56 is our structural spine for this, and we're going to layer in the clinical precision of Joblitz's Medical Microbiology, Chapter 42, just to flesh out the clinical picture and give you a whole story.
Our mission today is to build a mental model of this thing for you. Because if you are a medical student listening to this, or just someone fascinated by how fragile our biology actually is, prions represent a total glitch in the matrix.
A total glitch.
We need to understand how a simple protein, just a string of amino acids, something you eat every day, can turn into a brain-destroying monster.
And not just the how, but the why regarding detection. Because unlike a bacteria you can grow in a petri dish or a virus you can run a PCR on, these things are invisible to standard tests. If you don't know specifically what you are looking for, the clinical patterns, the timeline, you will miss it.
And missing it is fatal. Let's be real. There is no coming back from a prion disease.
None.
So let's start at the very beginning, the biological rule breaker. When the concept of a prion was first proposed, the scientific community didn't just embrace it, right? I mean, it sounded like magic.
Oh, they absolutely hated it. You have to understand the context here. In the mid-20th century, the idea that a protein alone could be infectious was heretical. It completely broke the rules. It was Stanley Prusiner who really championed the term prion.
Which stands for proteinaceous infectious particle, right?
You got it. He eventually won the Nobel Prize for it. But for a long time, people thought he was chasing ghosts. They were convinced there had to be a slow virus hidden somewhere inside that protein clump.
But there wasn't. They looked and looked, and no genetic material.
No, it really is just a protein. And Murray Chapter 56 makes a very specific distinction here that helps set the stage for you. Unlike a bacterium, which is a complex cellular machine with a cell wall and ribosomes, or a virus, which is genetic data wrapped in a delivery shell, a prion is hydrophobic.
It repels water.
Right. It forms clumps. And this is the part that keeps microbiologists up at night. It is highly resistant to protease.
Okay, let's define that for the non-chemists or the tired mid-students listening. Protease is the enzyme our body uses to clean up trash, right?
Exactly. Think of your cells as a busy commercial kitchen. You are constantly cooking up new proteins to do jobs. Structural beams, enzymes, signaling molecules. But you also make mistakes, or proteins get old and fold weirdly.
So you need to clean up.
Right. Protease is the garbage disposal in the sink. It grinds up the waste so the amino acids can be recycled. But a prion, it's like throwing a diamond into the garbage disposal.
Oh, wow.
It doesn't break. It just rattles around and eventually breaks the disposal.
I really like the conceptual anchor we discussed earlier, the zombie protein. It feels like the most accurate way to visualize this. Because zombies don't have babies, right?
It is the perfect analogy. Because usually, pathogens reproduce by making babies. A bacterium divides by binary fission. A virus hijacks a cell to print copies of itself. A prion reproduces by conversion. Imagine you have a room full of normal, healthy people. Then one zombie walks in. The zombie doesn't give birth to a baby zombie. It bites a human. Now the human becomes a zombie.
So the population of bad guys grows, but the total number of people in the room stays exactly the same. They just change sides.
Precisely. The prion recruits your own healthy proteins and turns them to the dark side. It is a corruption of the host's own supply. And that is why it is so terrifying. It's not an invader in the traditional sense. It's a mutiny. It's your own body turning against you.
That sets the stage perfectly. Let's get into the weeds of this mutiny. Section 1, structure and physiology, the Jekyll and Hyde nature of this protein. And this is a key point from the text. We all have the prion protein in our brains right now. I have it, you have it, the listener has it.
We do, and we actually need it. This is where the terminology gets critical for anyone taking a board exam. We have the citizen protein, which we call PrPᶜ, and that stands for cellular prion protein.
Exactly.
Okay, so where does this citizen live? It is anchored to the cell membrane of your neurons. Specifically, it uses a GPI anchor. Think of it like a little glycolipid hook. It uses that to tether itself to the outside of the cell, and it's found in very high concentrations in the brain, which gives us a big clue as to why the disease is purely neurodegenerative. It's practically coating the neurons.
So sitting on the surface of our brain cells, what is it doing there? I mean, it's not just decoration.
The jury is still somewhat out on its complete function. Biology still has its mysteries, but Murray suggests it plays a role in signaling. It modulates membrane proteins. It interacts with potassium channels and NMDA receptors. It might even help with copper transport.
So it's a functional, normal part of your neural architecture.
Yeah, it's a good citizen.
And structurally, if we could shrink down and look at PrPᶜ, what does it actually look like? Because this shape is super important.
This is probably the most important visualization of the whole deep dive. PrPᶜ, the normal protein, is rich in alpha helices.
Alpha helices, I always picture those like a slinky. Nice flexible coil.
That is the perfect image. A slinky, it's a spiral. Because of that shape, it is flexible. It is soluble, meaning it dissolves happily in water or cellular fluid. And most importantly, because it's open and flexible, it is protease sensitive.
So the garbage disposal can grind it up.
Right. If a protease enzyme comes along and snips it, the slinky just falls apart. It is completely recyclable.
Okay. So that is Dr. Jekyll, the nice, normal, soluble, recyclable slinky. Now, enter the villain, Mr. Hyde.
The villain is PrPＳC. The SC stands for scrapie, which is the prion disease in sheep.
Oh, because that was the first one described.
Exactly. But we use PrPSC as the generic term for the bad version in humans, too.
Now, does PrPSC have a different chemical formula? Is that a different set of atoms? Like, did it pick up a radioactive isotope or something?
No, and this is the mind-bending part. In sporadic cases, the amino acid sequence, the primary structure, is identical. It is the exact same atoms in the exact same order.
Wait, so it's the exact same ingredients, just cooked differently?
Folded differently. PrPSC has undergone a massive conformational change. Instead of those nice, flexible alpha helices, the protein refolds into beta sheets.
Beta sheets. That sounds a lot less fun than a slinky.
Think of a beta sheet like a sheet of paper that you've crumpled into a fan, or a pleated skirt, or even a piece of corrugated cardboard. It is flat, it is rigid, and it likes to stack.
Like stacking paper.
Exactly. If you have one sheet of paper, it's flimsy. If you stack 1,000 sheets of paper, you have a phone book. It becomes incredibly dense and hard.
So we've gone from a flexible slinky to a stiff stacked phone book.
You got it. And this shift from alpha to beta is the molecular smoking gun. Because of those beta sheets, the actual physics of the protein changes. It becomes hydrophobic. It hates water, so it clumps together with other beta sheets to hide from the cellular fluid. And crucially, it becomes protease resistant.
The diamond in the garbage disposal. The armor we talked about.
Yes. The enzymes that normally chew up proteins simply cannot find a grip. The beta sheets are stacked so tightly, like a closed book, that the protease scissors literally can't get in to cut the bonds. So the cell cannot break it down. It becomes an indestructible piece of trash in the brain.
So we have the setup. We have the slinky, which is PrPC, and the stiff sheet, which is PrPSC. Now, explain the mechanism of the bite. How does the sheet turn the slinky into another sheet? This is what Murray calls template-mediated protein refolding, right?
Right. This is a kinetic phenomenon. Prions replicate purely by contact. Imagine a clump of that abnormal PrPSC. It's floating along the cell surface, and it bumps into a normal PrPC.
The zombie bumps into the human.
But it's more than just a bump. It's a thermodynamic trap. The PrPSC acts as a template. It physically interacts with the alpha helices of the normal protein and actually lowers the energy barrier for them to flip. It forces the normal protein to unfold and refold into that abnormal beta sheet shape.
It just physically twists it.
It twists it. And now that newly corrupted protein isn't soluble anymore. It snaps onto the existing clump. It joins the zombie horde. And now that clump is slightly bigger, providing even more surface area to recruit more normal proteins.
It's an exponential cascade. It's like a crystal growing in a supersaturated solution.
It is exactly like that. You start with 1, it converts 1, now you have 2 templates, those 2 convert 2 more, now you have 4, then 8. It starts very slow. This is the long incubation period, but once the ball gets rolling, it becomes an absolute avalanche.
And this is all happening right on the surface of our neurons.
On the surface, yeah. And eventually, these clumps break off and accumulate inside the cell, clogging up the lysosomes, those are the cell's recycling centers, or they accumulate in the extracellular space as giant plaques. But the key takeaway for you here is that no new protein is being synthesized from scratch to cause the disease. The pathogen is made of you.
That is such a wild concept to wrap my head around. It completely challenges the definition of self versus non-self. Usually an infection is an invasion of a foreign army. This is a civil war.
A civil war where the rebels wear the exact same uniform as the loyalists, but they just march in a different formation.
Which leads us perfectly into section 2, pathogenesis. Because if you have all these stiff, indestructible sheets stacking up in the brain, the physical result is totally devastating. We call it spongiform change.
Spongiform encephalopathy. It's a mouthful, but the name is incredibly literal. Sponge-like brain disease.
So if I'm a student looking at a slide under a microscope, and for the listeners, this is a very classic image question on the boards, what am I actually seeing on that slide?
You're seeing vacuolation. Murray describes this very clearly. You see actual bubbles or holes inside the cytoplasm of the neurons and in the neuropil. The neuropil is just the mesh of axons and dendrites between the cell bodies. The brain tissue literally develops cavities.
Like Swiss cheese or a kitchen sponge.
Exactly. And as the neurons die from this massive accumulation of trash, you see a secondary reaction called gliosis. The glial cells, astrocytes specifically, are the support staff of the brain. When they see damage, they proliferate to try and patch things up. So you see this dense, fibrous scarring of astrocytes trying to hold the dying tissue together.
And what about the clumps themselves? Can we see the actual piles of beta sheets on the slide?
Often, yes. The PrPß aggregates form amyloid plaques. These are basically insoluble rods or fibrils. If you stain them with Congo red dye and look under polarized light, they show a very characteristic apple green birefringence. These plaques are basically graveyards of protein that the brain couldn't clear away.
Now, I really want to pause on something that I found baffling when I first studied this. You have holes in the brain. You have neurons dying by the millions. You have piles of protein trash everywhere. The immune system must be going absolutely haywire, right? I mean, if I get a tiny splinter, my finger swells up, turns red, gets hot. This is massive brain damage.
You would think so. You would expect a massive, raging encephalitis. But this is the immunological ghost concept. And it is a massive high-yield concept for exams because it is so counterintuitive.
Lay it on us.
There is no inflammation.
Not at all.
Zero. No neutrophils, no lymphocytes, no swelling, no fever.
That is terrifyingly silent. Why? Why is the immune system just sleeping through the apocalypse?
Go back to the structure. What is the PrPß actually made of?
Well, it's made of my protein. The amino acid sequence is exactly the same as the healthy one.
Exactly. T cells and B cells are trained in the thymus and bone marrow to recognize non-self sequences. They scan the amino acid code. When an immune cell bumps into a prion, it checks the ID card. It sees the sequence. It says, oh, that's just PrP. He works here. Let him pass.
It doesn't notice that PrP is holding a knife and wearing a scary mask.
The immune system generally doesn't check for shape. It checks for sequence. So the prion is totally invisible. It's hiding in plain sight.
This has to have huge diagnostic implications for us.
Huge. It means you have no interferon production. You have no antibody response. If a student sees a question asking about anti-PrP titers or serology for CJD, those are distractor answers. They are absolutely wrong. You cannot diagnose CJD with a blood antibody test because the body never makes antibodies against it.
That is a crucial point to highlight. If you're looking for an immune response, you are looking for the wrong disease entirely.
Correct. If you see inflammation on a test question, think viral encephalitis, think bacterial meningitis. Do not think prion.
Let's talk about the timeline here. We mentioned earlier that it starts slow. How slow are we talking?
Prion diseases were historically classified as slow virus infections, even though we obviously now know they aren't viruses. The incubation period is staggering. It can be months, years, or even decades. In Kuru, which we'll discuss in a bit, cases appeared up to 30 years after the cessation of cannibalism.
30 years. So you could be infected in your 20s and not show a single symptom until your 50s.
Yes. And during that entire time, the replication is happening silently. The beta sheets are slowly recruiting the alpha helices. The zombie count is rising, but it hasn't reached the tipping point yet to cause catastrophic cell death.
It's just a silent, decades-long buildup.
Jawob actually has an interesting detail here about the route of entry. If you ingest a prion, say, from contaminated meat, it doesn't go straight to the brain. It accumulates in the lymphoid tissue first.
Wait, I thought the immune system ignored it.
It does ignore it, meaning it doesn't attack it. But the prions can physically get stuck in the body's filters. You find them in the tonsils, in the Peyer's patches of the gut, and in the spleen. They use these lymphoid tissues as a staging ground. From there, they likely hitch a ride up the sympathetic nerves. They travel up the axons, retrograde like a climber going up a rope, until they hit the spinal cord and then finally the brain.
That journey up the nerve fibers must take a ton of time.
It takes a very long time. It can take years to climb that rope, but once they reach the cortex, once the sponge holes start forming, the progression shifts gears entirely. It goes from a slow crawl to a full-on sprint. Death usually occurs within months of the first clinical symptom.
So it's decades of absolute silence, and then you just fall off a cliff.
Exactly.
Okay, moving on to section 3.
Yeah.
Epidemiology. How do you actually get this? We've hinted at it, but let's break it down properly. Murray categorizes them into 3 distinct buckets: sporadic, genetic, and acquired.
Let's start with the biggest bucket. Sporadic CJD, Creutzfeldt-Jakob disease.
This is the most common form, right?
Well, common is a very relative term here. We are talking about 1 case per million people per year. So if you live in a city of 2 million people, you might see 2 cases a year. But of all the prion cases we do see, sporadic CJD accounts for about 85 to 90% of them.
And the cause for sporadic.
Bad luck.
Really?
Well, yeah.
Just random cosmic bad luck.
It appears to be a purely stochastic event, a random, spontaneous misfolding. Maybe a single protein flipped to the beta state by accident due to thermal fluctuation, or maybe a somatic mutation occurred in just one neuron that changed the stability of that specific protein. Once that first seed is formed, the chain reaction begins.
And who does this usually happen to?
It's overwhelmingly a disease of aging, usually people over 50 or 60. If you have a clinical vignette of a 65-year-old with rapid dementia, you're thinking sporadic CJD. It takes a lifetime for that random chance to occur or for the accumulation to finally reach a tipping point.
Okay, that makes sense. Bucket number 2, genetic or inherited.
This is about 10 to 15% of cases.
Mm-hmm.
These are families that carry a germline mutation in the PrPᶜ gene, which is located on chromosome 20.
So they're slinky is defective straight from the factory.
Exactly. The mutation might substitute one amino acid for another in a way that makes the alpha helix just a bit unstable. It doesn't guarantee it will flip today, but it makes it much, much more likely to flip spontaneously over time. It essentially lowers the energy threshold for the zombie conversion.
And what are the names for these genetic syndromes?
Familial CJD is the generic term, but there are 2 specific named syndromes you really should recognize for exams. One is Gerstmann-Sträussler-Scheinker syndrome, or GSS. This one presents with more cerebellar signs, ataxia, earlier on in the course.
And the other one?
The other one is the stuff of actual nightmares. Fatal familial insomnia, FFI.
FFI. That sounds like a horror movie title.
It is horrific to witness. In FFI, the prions selectively destroy the thalamus. Now, if you remember your neuroanatomy, the thalamus is the relay station, but it's also the major gatekeeper of sleep. It regulates your sleep-wake cycle. These patients completely lose the ability to enter deep sleep.
So they're just awake until they die.
They suffer from progressive, intractable insomnia. It leads to severe hallucinations, panic attacks, delirium. They eventually die from a combination of exhaustion and neurodegeneration. But structurally, despite the bizarre symptoms, it's the exact same underlying mechanics. Misfolded PrPᶜ.
Wow. Okay, bucket number 3, the one that gets all the media headlines, even though it's the rarest form, acquired infection.
This is where we talk about transmission. Prions as an actual infectious agent from outside the body. And we have to start with the incredible story of Kuru.
The Kuru story is fascinating medical anthropology. It really puts the culture in agriculture, or I guess not agriculture.
It happened in the Fore tribe in the highlands of Papua New Guinea. Back in the 1950s, researchers noticed the tribe was suffering from a mysterious, fatal, trembling disease. Kuru literally means shivering or trembling in the Fore language.
They thought it was genetic at first, right? Because it seemed to run in specific families.
They did, but the epidemiology just didn't make sense for a genetic disease. It was killing way too many people, and it was affecting adult women and children of both sexes, but rarely adult men. If it were X-linked or autosomal, the inheritance pattern was all wrong.
So why that specific demographic? Why women and children?
It turned out to be ritual cannibalism. When a relative died, the tribe would consume the body as a sign of deep respect to return the life force of the deceased to the hamlet. It was a sacred funeral rite.
And the Jalwet's text has a very specific detail on who ate what parts of the body.
Right. The men, being the warriors and hunters of the tribe, were given the choice cuts, the muscle meat. The women and children were responsible for preparing the body and were often left with the less desirable parts, which included the viscera and, crucially, the brain.
And the brain is where the prion load is the absolute highest.
Millions of times higher than in muscle tissue. They were handling the highly infectious brain tissue and ingesting it. They were basically inoculating themselves with massive doses of PrPß.
And once the practice was stopped in the late 50s, the epidemic slowly ended.
Very slowly. Because of that massive incubation period we talked about, new Kuru cases kept popping up into the 90s and even the early 2000s from infections that happened decades prior.
Then we have iatrogenic CJD. This translates to doctor-caused CJD.
This is the scary category for any medical professional listening. This implies transmission via medical procedures. Since we know standard sterilization doesn't work well on prions, we accidentally spread it before we knew better.
How exactly?
Contaminated neurosurgical instruments were a major vector. Prions stick to surgical steel like glue. Also, EEG electrodes implanted directly in the brain. Corneal transplants, prions accumulate heavily in the eye tissue.
And a really big one from history, human growth hormone, HGH.
Yes. Before we could make growth hormones safely in a lab using recombinant DNA and bacteria, we used to harvest it directly from the pituitary glands of human cadavers. They would pool thousands of pituitary glands together in giant batches to extract enough hormone.
Oh, wow. So if just one of those thousands of cadavers had undiagnosed CJD,
the whole batch was contaminated. Children who received those HGH injections for growth issues developed iatrogenic CJD years later. It was a massive tragedy, but it taught us a lot about the severe risk of pooled biological products.
Finally, in the acquired bucket, we have variant CJD, or VCJD, and the link to mad cow disease.
BSE, bovine spongiform encephalopathy. This was a massive epidemic in cattle, particularly in the UK in the 80s and 90s. It was caused by industrial farming practices. They were feeding cattle meat and bone meal, which contained the remains of other animals, specifically sheep that had scrapie.
Cows are herbivores. We essentially turn them into carnivores and cannibals.
And in doing so, we forced the sheep prion, scrapie, to jump the species barrier to cows, creating BSE. And then, tragically, it jumped the species barrier again to humans who ate the contaminated beef, specifically neural tissue that got mixed into the meat processing like hot dogs or burgers. This human form is what we call variant CJD.
Now, this leads to a really critical distinction. For the listener preparing for an exam, how do you actually tell the difference between a classic sporadic CJD case and a variant CJD case in a vignette? Murray and Jowitz are both very clear on this.
There are two main differentiators you need to look for: age and presentation.
Okay, let's break down age first.
Sporadic CJD is a disease of the elderly. The average age of onset is around 68. Variant CJD, on the other hand, affects much younger patients. We are talking 20s and 30s. The average age is often cited as 28.
That is a massive difference. If I see a 25-year-old with CJD symptoms on a test, it's almost certainly not sporadic.
Highly unlikely. That would be a medical anomaly on top of an anomaly.
And presentation, how do they differ there?
Sporadic CJD presents with dementia and motor signs first. Memory loss, confusion, ataxia. Variant CJD often starts with psychiatric symptoms: severe anxiety, depression, social withdrawal, bizarre behavioral changes.
So a 25-year-old presenting with sudden severe depression and weird sensory feelings.
That is a huge red flag for VCJD, especially if there's a history of residence in the UK during the 90s. The patient often ends up in a psychiatry clinic first, getting treated for depression or psychosis before the severe neurological decline actually begins.
Got it. So old and forgetful equals sporadic. Young and depressed or anxious equals variant.
That's the perfect high-yield heuristic to remember.
Let's move to section 4. Clinical syndromes. We need to recognize the pattern when it walks through the door, or, more likely when it's wheeled in. What is the classic CJD presentation?
We look for a classic triad: And you absolutely need to memorize this triad. First is rapidly progressive dementia.
Contrast that rapidly with something like Alzheimer's.
Alzheimer's is a slow burn. It takes years. Families adjust to it gradually. They say, oh, grandma's getting a little forgetful. CJD is terrifyingly fast. We are talking forgetfulness progressing to profound dementia, mutism, and coma within just a few months. Families are often totally shocked by the speed. They'll tell you he was fine at Christmas, and by Easter, he didn't know who we were.
Second part of the triad.
Cerebellar signs. This is the ataxia. Staggering gait. Profound clumsiness. Difficulty speaking, which is dysarthria. The cerebellum is full of granule cells that get hit incredibly hard by the spongiform change.
And the third, I know this is a huge buzzword.
Myoclonus.
Let's find that for us.
Muscle jerking. But specifically startle-induced myoclonus, if you clap your hands loudly next to the patient's ear, or slam a door, or shine a bright light in their eyes, the patient will jerk or twitch uncontrollably. It's a massive, hyperexcitable reflex.
So rapid dementia plus ataxia plus startle myoclonus.
That is your classic CJD triad.
Let's construct a mental sandbox case to lock this in.
A vignette.
Okay, let's do it. You have a 68-year-old retired librarian.
Age-fit sporadic.
She comes in because her husband says she's been getting lost inside her own house. It just started 2 months ago, and now she can't even feed herself.
Extremely rapid progression.
On physical exam, she has a wide-based gait and severe intention tremors. During the exam, you accidentally drop your metal clipboard on the floor, and her whole body violently jolts. The startle myoclonus.
Exactly. That is a slam dunk case for Creutzfeldt Jakob disease.
And the prognosis for her.
Grim. Death usually occurs within 6 to 12 months of symptom onset. Usually from aspiration pneumonia because they completely lose the ability to swallow and protect their airway.
Let's assume we suspect it based on the triad. How do we actually prove it? Section 5. Diagnosis. Hunting the invisible.
This is incredibly frustrating clinically. You cannot culture a prion. You can't put it on a blood agar plate and see what grows. You can't run a standard PCR for DNA because there is no DNA. You can't look for antibodies in the blood because there are no antibodies.
But what do we actually do?
The gold standard is still brain biopsy or autopsy. You have to look at the brain tissue under a microscope and see the spongiform changes and use immunohistochemistry to stain for the PrP plaques. But obviously, a brain biopsy on a living patient is highly invasive and very risky.
So what are our premortem proxies? How do we diagnose it while the patient is still alive?
MRI is very helpful. You look for hyperintensity's bright spots in the basal ganglia, specifically the caudate imputamen and the cortex. It's often called cortical ribboning. It looks like a bright ribbon tracing the outer edge of the brain on the scan.
And the CSF. Can we do a lumbar puncture?
We can. And here we look for markers of rapid brain destruction. The classic one mentioned in Murray is the 14-33 protein.
14-33, is that the prion itself?
No, that is a very important distinction to make. 14-33 is a totally normal intracellular protein that lives inside healthy neurons. It should not be floating free in your spinal fluid. If you find it in the CSF in high amounts, it means neurons are exploding and leaking their contents.
So it's a marker of, hey, brain cells are dying rapidly.
Exactly. It's supportive evidence, but it's not specific to prions. You could see elevated 14-33 in a massive stroke or severe viral encephalitis too. Tau protein is another one you can look for. It also indicates massive neuronal damage, but these are just the smoke. We want to see the fire.
Right. Is there any test that actually signs the prion itself while they're alive?
There's a newer test that Murray highlights. It's an absolute breakthrough. It's called RT-QIC.
RT-QIC, real-time quaking-induced conversion. That sounds like a seismology term.
It sounds super cool, and it actually uses the prion's own superpower against it. Remember how the prion converts normal protein into bad protein by contact?
The template effect, the zombie bite.
Right. So in the lab, they take a tiny sample of the patient's CSF, or sometimes even a nasal brushing from the olfactory epithelium. They mix it in a test tube with a large supply of normal recombinant PrP protein, and then they literally shake it. Quaking.
They shake it to speed up the contact between the proteins.
Exactly. Kinetic energy. If there is even a tiny microscopic amount of PrPß in the patient's sample acid, it will start converting the normal protein in the test tube into beta sheets.
It starts the chain reaction right there in the tube.
Yes. And as the new aggregates form, they use a special fluorescent dye called thioflavin T that specifically binds to beta sheet clumps. So if the tube starts glowing under the light, it means the infectious seed was there.
That is incredible science. using the zombie bite mechanism to detect the zombie.
It's highly sensitive and very specific. It's becoming the absolute game changer for diagnosis because it detects the actual pathogen's activity, not just the bystander brain damage.
Okay, we've diagnosed it. Now the bad news. Section 6, treatment.
It is entirely bad news. Therapeutic nihilism is the phrase often used here. There is no treatment. No antiviral, no antibiotic, no immunosuppressant works. We can only offer supportive care to make the patient as comfortable as possible.
The focus then has to shift entirely to prevention and disinfection. And this brings us back to the terminator nature of this protein.
This is an exam-critical section. Pay attention. You will get a question on how to clean surgical instruments used on a suspected CJD patient.
Can I just boil them?
No. Prions easily survive boiling water.
What about chemicals? Can I use formaldehyde?
Absolutely no. Prions love formaldehyde. In fact, formalin fixes tissues by cross-linking proteins, which might actually stabilize the prion amyloid. It essentially embalms the prion, keeping it fresh and infectious. Never use formalin.
What about radiation?
Nope. UV light and ionizing radiation kill things by damaging nucleic acids, breaking DNA or RNA. Prions don't have DNA, so radiation is completely useless. It's like trying to kill a rock with a sun lamp.
What about the standard autoclave? The usual 121 degrees Celsius for 15 minutes that kills all bacteria and tough spores.
Still not enough. The beta sheet aggregates are incredibly heat stable. Standard autoclaving might not kill them completely. There have been documented cases where depth electrodes were autoclaved, reused on a new patient, and still transmitted CJD.
Wow. So what is the nuclear option? What do I actually have to do to clean these tools?
You have two main weapons. First, heat, but it has to be extreme heat. Murray specifies 132 degrees Celsius to 134 degrees Celsius for 1 full hour, not 15 minutes, an entire hour.
1 hour. That is a massive cycle.
Or you use chemical warfare. You need something that hydrolyzes or denatures protein structure very aggressively. You use 1 normal sodium hydroxide, which is NaOH, basically lye, a very caustic base, or 5% hypochlorite, which is undiluted industrial bleach.
So basically, you just have to chemically melt the protein structure apart.
Yes. You usually soak the expensive surgical instruments in the base for an hour, and then you autoclave them on the extreme cycle. And because this protocol is so incredibly damaging to the instruments, it literally corrodes surgical steel. The reality in many modern hospitals is that if instruments are used on a known CJD patient, they are simply incinerated. They are thrown in the fire and destroyed.
That really drives home how resilient this thing is. It's just a single molecule, but it's tougher than life itself.
It really is the most robust pathogen we know of. It persists in soil for years. It survives the digestive tract acids. It survives the standard autoclave. It demands immense respect.
So let's wrap this up. This has been a pretty heavy deep dive.
It really is. We've covered the biological rule breaker, a pathogen with absolutely no DNA.
You unpack the structure. The slinky, which is PrPC, versus the sheet, which is PrPßE, alpha to beta, soluble to insoluble.
We've looked at the pathogenesis, the template-mediated refolding trap, the spongiform holes in the brain, the profound immunological silence.
We've broken down the epidemiology. Sporadic, genetic, and acquired like Kuru, iatrogenic cases in mad cow.
And finally, the sheer impossibility of killing it. The absolute need for extreme heat and harsh chemicals to prevent iatrogenic spread.
It's a lot to take in. But before we sign off, we need to test your recall. It's time for the deep dive rapid fire quiz.
I'm ready. I'll read the scenario. You, the listener, take a second to think, and then I'll give the answer and the why.
Let's do it.
Question 1. A neurosurgeon performs a brain biopsy on a patient who turns out to have sporadic CJD. The scrub nurse asks how to sterilize the instruments. Which method is required? Option A, boiling for 30 minutes. Option B, soaking in 70% ethanol. Option C, standard autoclaving at 121 degrees Celsius for 20 minutes. Option D, immersion in 1 normal sodium hydroxide or autoclaving at 132 degrees Celsius for 1 hour.
Lock in your answer.
The answer is D. Prions are incredibly resistant to boiling, alcohol, and standard autoclaving. You need the extreme measures. 1 hour at 132 degrees or sodium hydroxide. This is a classic safety question you will see.
Got it. Extreme heat or lie? Next question.
Question 2. You are examining a histology slide from the brain of a patient who tragically died of Kuru. What is the hallmark finding? Option A, Negri bodies. Option B, neutrophilic infiltrate. Option C, spongiform vacuolation of neurons without inflammation. Option D, paravascular lymphocytic cuffing.
Think about the immune response or lack thereof.
The answer is C, spongiform vacuolation without inflammation. Remember, holes but no heat. Negri bodies are rabies, which is from Jawitz chapter 42. Neutrophils and lymphocytes imply an active immune response, which prions simply do not trigger.
Holes but no heat. I really like that mnemonic. Let's say question 3.
Question 3. Theoretically, what is the fundamental molecular difference between the normal cellular prion protein, PrPᶜ, and the pathogenic scrapie protein, PrPＳC? Option A, PrPSC contains RNA. Option B, PrPSC has a completely different amino acid sequence. Option C, PrPSC has a high beta sheet content and is protease resistant. Option D, PrPSC is highly soluble in water.
Think about the shape.
The answer is C. It is a conformational change. Alpha becomes beta. Slinky becomes sheet. This creates the protease resistance. The amino acid sequence is identical.
It's all about the fold. Question 4.
Question 4. A 28-year-old male presents with severe anxiety, depression, and odd sensory sensations. Months later, he develops ataxia and dementia. He lived in the UK during the 1990s. This presentation is most consistent with option A, sporadic CJD, option B, variant CJD, option C, Gerstmann-Sträussler Scheinker syndrome, option D, Kuru.
The age is the big clue here.
The answer is B, variant CJD. The keys are the age, young at 28, and the presentation, psychiatric symptoms first. Sporadic is old with dementia, variant is young with psych symptoms, and the UK history strongly links to the BSE mad cow epidemic.
All right, last one. Bring it home.
Question 5. Which finding in the cerebrospinal fluid is most suggestive of Creutzfeldt-Jakob disease? Option A, elevated protein 1433. Option B, high neutrophil count. Option C, low glucose. Option D, presence of anti-PrP antibodies.
Remember the proxies.
The answer is A, elevated 1433. This is a non-specific marker of rapid neuronal destruction used in diagnosis. Neutrophils and low glucose would be bacterial meningitis. Antibodies are entirely absent in prion disease.
Perfect. How did you do? Hopefully you nailed those.
If you want to really solidify this, go read Murray chapter 56. It's short but incredibly dense. Look at the diagrams of the alpha to beta conversion. That specific image should be burned into your retina for exam day.
And as a final thought to leave you with, just think about the sheer fragility of life. We're entirely built of proteins. We rely on them folding into the absolute perfect shapes to let us breathe, to think, to love. And all it takes is one single protein folding just slightly the wrong way to bring the whole house of cards down.
It's a humbling reminder that structure is function, and sometimes shape is destiny.
Thanks for joining us on this deep dive. We'll see you next time.
Stay curious out there.